In this video, we are covering a new neural network architecture for image segmentation called UNet. This architecture was initially developed for biomedical images, but like many other fields, the same technique can be applied to any kind of segmentation. In fact, biomedical images are very hard in some sense compared to regular segmentation tasks like people segmentation. This was inspired by the FCN network, which we saw in the last video. And one of the characteristics of the UNet design is that it is approximately symmetric. If you remember the FCN structure, it was not symmetric. It was on one side, on the encoder side, we had used the standard backbone for image classification. On the decoder side, it looked very different than the encoder. But UNet looks similar on both sides. The second difference between FCN and UNet is that it learns the boundaries of the same class also. So the loss function is slightly different so that it is able to learn the boundaries of the same class. And this is important because in a lot of biomedical images, they have examples where two cells are touching each other. And we will see this in the next few slides. And therefore, it is important to separate those as two different objects. In that sense, this is more like instance segmentation instead of semantic segmentation. And then they use a tiling strategy that allows them to process very large images. And that's also because in biomedical image processing, the image sizes are usually very large. When I was in grad school, I used to work with images in biomedical image processing. These were cryo-electron microscopy images, and these images were 6K by 6K images 20 years back. So now I don't even know what the sizes of these images would look like. So it is important that we develop algorithms that can be applied to very large images. And that is why they use a tiling strategy. The other problem with applying computer vision to some of these biomedical problems is the lack of training data. In this paper, the researchers had access to only 30 segmented images. So you have to use data augmentation very heavily in such applications. Now let's look at the architecture. Now it becomes immediately clear why this is called UNet. So you can see that the architecture looks like a U. And these images have been copied from the original paper. I really like how they have represented the architecture in this way, but it requires certain explanation. First of all, we have the input image here, and the input image size is 572 by 572. If it is a larger image, you can select a tile from inside the input image and do this calculation. Now, you can also see that the image has only one dimension. Now, what they have done is on the side, they write the spatial dimension, and on the top, they write the number of channels. This is actually a very good way of representing the network. And these arrows mean convolution, and they are using only one kind of convolution. It is 3x3 convolution, followed by ReLU activation. And they also use this max pooling layer, which downsamples the image by 2. So the initial 568 by 568 becomes 284 by 284. At the same time, in the same level, they increase the depth of the network by 2. So here you can see that the depth of the network or the number of channels of the tensor was 64. And here they have increased it to 128. And as you go deeper into the network, the depth increases by 2, and the width and height, they decrease by 2. So you can see along this. This is also very symmetric in the sense that once you downsample the image, you're reducing the spatial dimension of the image, but at the same time, you're getting more and more confident about what this object is. So the information about where is lost, but the information about what increases as we go down the network. And we have discussed this before also. And in case of classification, the where is completely gone and only the what remains. In this network, the size goes to 32 by 32. So we still have some wear information and a lot of watt information. The other thing they do is use the same kind of upsampling mechanism as we had seen in FCN. So they use the deconvolutional layer to do this upsampling. The green arrows show upsampling. And instead of adding the information that we get from different layers, they concatenate the information. So the volume that they get from upsampling this layer, they concatenate to that volume additional information from this layer. And because they are of the same size, we can do that concatenation and keep doing the upsampling. So idea-wise, it is very similar to FCN. The idea is that we are first downsampling the image and then upsampling it back again. And this looks like a U. And while upsampling, we are also merging information from higher layers, like here. These gray arrows show that. So that's the basic architecture. You can see that the segmentation map is of size 388 by 388. It is smaller than the size of the input image, and that is why they have to use tiling to fix the problem. So they have to apply this method multiple times over the image and cover all the holes. The other thing to note is that the output segmentation has two channels, and this is because there are only two classes. One is the object, whatever they want to detect, and the other is the background. Now let's go over what is overlap tiling. So this is basically a method to go window by window and do the segmentation in that window. You will get a smaller output, but that's okay. So for example, this blue window shows the region that is used to get this yellow output. And of course, there is a problem what happens at the boundaries. So the original image here is only this green region, the region we have shown using a green boundary. That is the original image. So at the boundary, you can see this is the yellow region, which corresponds to this output yellow region. However, the input region needs to be larger. What do we do? How do we get the data in these locations? We had seen in case of FCN, the strategy they had used for such things was they zero padded the image. However, in this paper, they decided to use a mirror reflection. So if you look at this region is a mirror reflection of this region. You can see these two structures. Similarly, this region is a mirror reflection of this region about this axis. And this region is a mirror reflection along this point. So you cover all these regions with kind of fake data, but that's fine for this purpose. Then you learn the mapping, you learn the output. And then you move this window sideways and create another map and fill out the whole image. So that's the idea of overlap tiling. And that is how they are able to cover the entire large image, which could be, 10K by 10K. Now, the second thing to keep in mind is that they had very little information in terms of data. They had only 30 images. So the data needed to be augmented. Whenever possible, we should augment the data. And whenever possible, we should use both linear and nonlinear augmentation. One simple example of augmentation is that you change the size of the image. You can also change the color of the image, and you can apply some affine transform to the image. However, when you have very little data and when we have this kind of biomedical images, it may also be possible to use nonlinear transformations, which are not like affine transforms, because this nonlinear image is still a valid example. So in this case, what they did was they applied a nonlinear map, and you can see the nonlinear map on this green image. The original image I have not shown here, but the original image was transformed to this nonlinearly mapped image, and the grid, shows that deformation. So from 30 images, they might have gone to a very large number of images by applying these deformations. And of course, the standard augmentation techniques like reflection, etc. still apply. So the other important change they made was in their loss function. And the reason they had to change the loss function is that in biomedical images, this is the input image that you're looking at. As you can see that this is a very difficult image, especially because all these regions, which are the foreground, they are touching each other. So if you look at the ground truth, which is shown in this, the various elements are touching each other. And if you just do standard semantic segmentation, you won't be able to tell the difference between this and this because they are technically the same object, and so there should not be any boundary between them. However, this is a different application, and they had to make sure that the results have the right boundaries. So they introduce this border wherever there was a boundary. So this is an artificial border, 1 pixel thick, which was introduced. And if this border is crossed, they had a huge penalty in the loss function. So this shows a visual representation of the loss function. And you can see that in the red regions, which correspond to the boundary, they have a huge penalty if this boundary is not respected. So that's how they controlled the segmentation process, by adding a boundary and penalizing whenever you cross the boundary. And this is a very standard way of, training a neural network. The things you don't want it to do, penalize it more than anything else, right? And the other reason this kind of loss function was very important is that the boundary pixels are way fewer than other pixels. So we have to increase the penalty when you breach the boundary because there are so few pixels. They will be overwhelmed by the rest of the image if you don't overcompensate for this boundary. Now let's go over some results. I want you to appreciate how difficult these results are. And because I have also worked in similar areas, these problems used to be extremely difficult to solve 10 years back. So we would not even have thought that, okay, we were just a few years away from having a solution. These used to be extremely difficult. And here you're looking at a result. There is some cell here. The ground truth is shown using yellow, and the results they obtained using UNet is shown in cyan. So very nice results. Similarly, in this case, you can see the ground truth in yellow and the results in a different color. So that is all we wanted to cover in this video. I hope you appreciate how difficult some of these problems are. They may not look very difficult to you now because they are solved, but coming from somebody who has worked on some of these problems in an earlier life, I have to say that it is so impressive to see these kinds of results these days. We did not imagine that we will be getting such great results very soon. It's even difficult for humans to do. The second thing I want you to appreciate is that the ideas from one field, if you take them and publish them in a different field, you can make a huge difference. The same ideas from one field, let's say you were working in semantic segmentation on regular RGB image, and now you took those ideas and applied it to a different field, it can be a revolution in that field. I want to just end with a little story. When I was in grad school, I was working in cryo-electron microscopy in addition to some other regular computer vision work that I did. And I took the Viola and Jones algorithm and applied it to detecting protein macromolecules in cryo-electron micrographs. And I was not very proud of that work because it was literally taking one algorithm and applying it to a different problem. And so I had a talk with my advisor that, this is not substantial work. How can we publish it? And he said that we are thinking too much about our own ego. This would be a very good result for the cryo-electron microscopy community. And as long as we clearly mention that we are taking an algorithm which is already published and applying it, it is for them to decide whether it's a good idea or not. So I reluctantly agreed. And we had clearly mentioned that this exact algorithm has been applied to face detection. And we took that idea and we are applying it to cryo-electron microscopy, images and detecting protein macromolecules instead. And that had such a positive response. That paper that I had published, that was selected to be the cover of Journal of Structural Biology for that edition in which the paper appeared. My paper was selected to be the cover of that edition. So the takeaway is that the things that you learn in one field, apply it to a different field, and it can be a revolution in that field. That is all I wanted to cover in this video. Thank you so much.